Welcome to G-Fun Facts Online. Today we are looking upward to solve a problem that has plagued medicine down here on Earth for decades. We are talking about a silent war being fought in laboratories against a fundamental force of nature, gravity. Specifically, we are exploring how the future of human tissue engineering and the cure for paralysis is currently orbiting 250 miles above our heads. To understand the magnitude of this breakthrough, we first need to look at the limitations we face on the ground. In the field of regenerative medicine, the dream has always been to engineer complex, functional human tissues to replace damaged ones. But on Earth, bioengineers are constantly hampered by physics. When they attempt to build a three-dimensional tissue structure in a terrestrial lab, they are forced to use scaffolds. These are synthetic or biological lattices designed to hold cells in place. They are necessary because, without them, gravity ruins everything. It causes the liquid bioinks, which are suspensions of living cells and nutrients, to collapse into a puddle before they can set. Cells sediment to the bottom of droplets, nutrients distribute unevenly due to convection currents, and delicate microchannels intended to guide nerve fibers sag and deform under their own weight. This limitation is particularly devastating for neural tissue engineering. You see, the human nervous system isn't just a bag of cells. It is a hyper-precise geometric arrangement. A peripheral nerve consists of bundles of axons, called fascicles, which are aligned with micron-level precision and encased in protective sheaths. If you try to print this architecture on Earth, the tiny channels required to guide regenerating axons, often as small as 10 to 50 microns in diameter, simply collapse. You end up with a structure that might look like a nerve on the outside, but fails to function like one on the inside. However, aboard the International Space Station, or ISS, this constraint vanishes. In the microgravity environment of low Earth orbit, the rules of biomanufacturing are effectively rewritten. Here, surface tension becomes the dominant force, overpowering gravity. Liquids can be manipulated into complex shapes without collapsing. Cells float suspended in their medium, interacting freely in 3 dimensions, just as they do during embryonic development. This is the dawn of orbital bioprinting. It is no longer science fiction. It is a commercial reality focused on manufacturing medical devices in orbit that are physically impossible to create on Earth. Let's dive into the physics of why this works, because it is fascinating. On Earth, printing a vascularized or channel-rich tissue requires a difficult trade-off. You can make the bioink thick and viscous to hold its shape, but this suffocates the cells by limiting nutrient diffusion. Or, you can make it thin and watery to keep the cells happy, but the structure collapses into a blob. In microgravity, this trade-off disappears. Because there is no sedimentation, meaning Stokes' law is functionally nullified, cells remain perfectly suspended in the bioink. This allows scientists to use ultra-low viscosity bioinks that are rich in nutrients and allow for rapid cell signaling. Furthermore, the absence of buoyancy-driven convection means that biochemical gradients remain stable. For nerve tissue, this is critical. Nerves grow by following chemical cues, a process called chemotaxis, and physical cues, known as contact guidance. In space, a bioprinter can extrude a hydrogel containing a specific gradient of nerve growth factor that remains perfectly stratified, creating a stable highway for axons to follow. The transition from petri dishes to industrial manufacturing in space has been driven by sophisticated hardware. 2 key systems exemplify this leap: the Biofabrication Facility, or BFF, by Redwire Space, and the Auxilium Microfabrication Platform, or AMP-1, by Auxilium Biotechnologies. The BFF was the first American system capable of manufacturing human tissue in microgravity using soft, ultrafluid materials. But the specific requirements of nerve tissue led to the development of the AMP-1, which was deployed with a singular focus: repairing peripheral nerve damage. In early 2025, Auxilium Biotechnologies achieved a historic milestone using this platform. They simultaneously manufactured 8 implantable medical devices known as NeuroSpan Bridges aboard the ISS. These bridges are designed to treat peripheral nerve injuries, severed nerves in the arms or legs resulting from accidents or combat. When a nerve is severed, the ends retract. If the gap is large, the standard of care on Earth is an autograft, where a surgeon harvests a sensory nerve from the patient's own leg to bridge the gap. This causes permanent numbness in the leg and has a high failure rate. While synthetic tubes exist on Earth, they are simple and hollow, often leading to painful tangles called neuromas. The space-manufactured NeuroSpan Bridge is different. Because it was printed in microgravity, it features internal microchannels, tiny, distinct tubes within the larger tube that mimic the natural fascicular architecture of a human nerve. On Earth, printing these 10-micron walls within a soft hydrogel would be impossible. They would sag and fuse. In space, they remain distinct, open, and perfectly aligned, acting as physical guardrails that force regenerating axons to grow straight across the gap. While the structural advantages are profound, the biological effects are perhaps even more startling. Research has revealed that neural stem cells behave fundamentally differently in orbit. In a landmark study, researchers found that human neural stem cells proliferated 7 times faster in space than identical control cultures on Earth. This isn't uncontrolled growth like cancer. It represents a preservation of stemness. On Earth, stem cells often age or differentiate prematurely due to mechanical stress. In microgravity, the lack of shear stress keeps them in a potent, youthful state for longer. Gene expression analysis has shown an upregulation of survival and proliferation genes in space-flown cells. Crucially, space-grown stem cells show a bias towards neuronal differentiation rather than astrocytic differentiation. This is vital because one of the biggest enemies in nerve repair is the glial scar, a dense wall of astrocyte cells that blocks regeneration. Space-based experiments show a decrease in astrocyte markers and an increase in neuron markers, implying that space-engineered tissues might be biologically tuned to regenerate neurons rather than forming scar tissue. We also see an accelerated maturation effect, often called the time machine effect. Brain organoids, miniaturized 3D models of the human brain, grow larger and more complex in microgravity without the dead, necrotic cores often found in Earth-grown samples. Features of neural development that take weeks on Earth can appear in days in orbit. This means a functional nerve graft could potentially be grown, matured, and ready for implantation in a fraction of the time required on Earth. The transition from research to therapy is already underway. Following the successful manufacturing run on the ISS, Auxilium Biotechnologies initiated the NeuroSpan clinical trial in 2025. This study aims to enroll 80 patients with traumatic nerve injuries, marking the first time a medical device manufactured in space has entered a regulated human clinical trial. If successful, this validates the economic model of orbital manufacturing, proving that the high cost of launch is outweighed by superior clinical efficacy. Looking forward, the holy grail of this technology is the repair of spinal cord injuries. Currently, these injuries are permanent because the central nervous system lacks the regenerative drive of the peripheral system. Orbital bioprinting offers a two-pronged solution: structural bridging using scaffold-free, highly aligned neural bridges, and cellular therapy using space-expanded neural stem cells primed to suppress scarring. Experiments are already being planned for spinal patches customized to a patient's specific injury geometry. As the ISS nears retirement in 2030, commercial space stations like Orbital Reef and Axiom Station are designing biomedical factories as core revenue streams. We can envision a future where a patient's stem cells are launched to orbit, a custom organ or nerve graft is printed and matured in an automated biofoundry, and the finished tissue is returned via a cargo glider for implantation. Of course, challenges remain. The high G-forces of re-entry can damage delicate biological tissues, requiring specialized cushioned capsules. Cosmic radiation is another concern, requiring careful shielding to prevent DNA damage in long-duration growths. And while the cost is currently high, the plummeting price of launch via reusable rockets is making the economic case increasingly compelling. Orbital bioprinting represents a fundamental decoupling of manufacturing from the constraints of our planet. For thousands of years, we have built things based on what gravity allows. Now, we are building based on what biology requires. As the first patients receive their space-manufactured nerve implants, we are witnessing a new industrial revolution, where the harsh vacuum of space is combined with the delicate complexity of life to heal injuries once thought permanent. Thank you for listening to G Fun Facts Online.